Welcome to Let's Get to the Marks, going through a Benedict's test and reducing sugars A-level biology question. Don't forget to pause the video, have a go, write your answer on scrap paper, then check the answers. Figure 2 shows the structure of two alpha glucose molecules. On Figure 2, draw a box around one chemical group in each glucose molecule used to form the glycosidic bond. So, where does the glycosidic bond form between these two glucose molecules? Well, a condensation reaction takes place. You've got to ask yourself, what gets lost or removed in a condensation reaction? Of course, it's H2O that's removed. But what two groups does it come from? It comes from these hydroxyl groups. The OH groups are called hydroxyl groups. So you could circle both of them, or you could just circle an H on one and an OH on the other. So either of these are correct and acceptable. The mark scheme's first point is to circle both chemical groups, which are the two hydroxyl groups. But of course, you only lose one OH and one H. So you could just circle an OH and then an H. 3.2. A precipitate is produced in a positive result for reducing sugar in a Benedict's test. A precipitate is a solid matter suspended in solution. A student carried out the Benedict's test. Suggest a method other than using a colorimeter that this student could use to measure the quantity of reducing sugar in the solution. So, once again, going back to this, a precipitate is a solid that forms in a liquid. So with the Benedict's test, you're using copper sulfate and sodium hydroxide, which has a blue color, and you get a red precipitate. So how could we, without using a colorimeter, how could we measure how much sugar was in there or get some sort of idea, a comparative idea? Well, we could filter off the solution, dry the precipitate carefully with paper towels, and then weigh it or measure its mass on a balance. And therefore, if you had two different solutions, the one with the higher mass of red precipitate is going to be the one that has the most sugar in it. So we could use that as a quantitative measure of the glucose concentration, or sorry, the concentration of reducing sugars in the solution. So what do you get the marks for here? One is for filtering the solution off and then measuring the mass. Right, let's move on. In an investigation, a student wanted to identify the solutions in two beakers, A and B. She knew one beaker contained maltose solution and the other beaker contained glucose solution. So let's label one of these M for maltose and one G for glucose. Both solutions had the same concentration. She did two separate biochemical tests on each beaker. Test 1, she used Benedict's test on each beaker to see if there's a reducing sugar. Test 2, she added the enzyme maltase and then repeated the Benedict's test at a temperature of 30 for 5 minutes. Now we can see that beaker A is the one with just the reducing sugar, and beaker B, or the glucose, and beaker B contains the maltose. Now what's happened with beaker B? It was red, but then it goes dark red. Why? Why in test 2 does beaker B go dark red? Well, we added maltase, and that's an enzyme. So let's find out. Explain the results for beakers A and B in the table. Well, the reason why beaker A goes red with both tests is because it shows the presence of glucose, because we've done the Benedict's test and glucose is a non-reducing sugar. However, with beaker B, it goes dark red on test 2 because the maltase, which is an enzyme, hydrolyzes the glycosidic bond in the maltose, snapping it into two glucose molecules. So if you have the same concentration of maltose and the same concentration of glucose, if you hydrolyze the maltose, you'll double the concentration of the glucose because each maltose molecule is made of two glucose molecules. So although you might think you have the same concentration of maltose as glucose, each maltose molecule is made of two glucose molecules, so you have double the concentration of glucose in a maltose molecule. It's made of 2G. Right. Let's move on to the next part of this question, as we have solved that one. So you get 2 marks here, which is nice. Don't forget to hit the like and subscribe, people. It really helps the algorithm, the YouTube algorithm, get my stuff out to other people. 3.4, use a colorimeter in this investigation, or use of, would improve the repeatability of the student's results. Why? So what does a colorimeter measure? It measures absorbance or you can set it to measure transmission. And that means instead of just looking and going, has that gone red? Is that dark red or is that medium red? You actually get a quantitative result. You get a number so you can judge and compare fairly two different concentrations. Whereas otherwise, you're using a subjective decision when saying, is this red darker than that red? That's down to people's eyesight and down to people's opinion. We call that a subjective result or evaluation. We don't really want that. So you get a quantitative result with a colorimeter. So in test one, the student used a measuring cylinder to measure 15 centimeters, that's the measurement, from a beaker. The measuring cylinder gives a volume that has an uncertainty of plus or minus 1 centimeter cube. She used a graduated syringe to measure 5 centimeters. So that's a second measurement using a different piece of equipment. So we have a measuring cylinder and graduated syringe. The graduated syringe gives a volume with an uncertainty of 0.5 centimeter, plus or minus. She mixed these volumes of liquid to do a biochemical test, calculate the percentage area for the measurements used to obtain the full 20 centimeter cube mixture of the solution from the beaker and the Benedict solution show you're working. We need to treat them as two separate, then add them together. So for our measuring cylinder, we had an uncertainty of 1. We're going to divide that by the measurement. So the percentage error is 1 over 15 times 100. So we can do that now, 1 over 15 times 100, to work out the percentage error of the measuring cylinder, or we can combine it with the percentage error of the graduated syringe. Now, the graduated syringe has an uncertainty of 0.5, and the measurement taken was 5 centimeters. So we're going to do, don't need the C there, we do 0.5 over 5. We can add these together if we like, and then we can times 100, and that's going to give us our answer. Dead easy. So if you do that, you get an answer of like 16.66 recurring. You could round that up to 16.67, but there's a whole range up to 17% they give you. And there we go, just a quick little video. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. Let's smash those exams.
You're still here? It's over.